Thank you very much. I'd like to first thank the Vice-Chancellor and the Dean, all of the Deans, and you, my colleagues and friends here from the university, and especially also a big thank you to my family, without which I would not have got to this part of my life, I think. So particular thanks to my husband, my children, and my parents, who are all here in the audience. And it's especially thanks to my parents who were enabled to model and show scientific thought and questioning the environment and creativity. And so I'm really delighted to be here, and thanks very much to them. And with that, I would like to share with you a little bit of a journey, a story, if you will, of fungal-like microbes and my love for these organisms. I've been working with them for about 20 years now. And why are they important? Because both fungi and fungal-like organisms hinder food security. And that's really a key that underpins my work, that diseases of plants have economic, social, and ecological implications, and that can lead to a huge impact on human health and well-being. And we know that the global population is growing and was estimated to be at or above 9 billion by 2050. And what we know also is that we need then to produce more food to feed those people. But diseases, particularly caused by fungi and fungal-like organisms, are major threats to that. If we look at the top 5 food crops in the world, that is rice, wheat, potatoes, maize, and soybean, per year, over 175 million tons of crops are lost to damage by fungi and fungal-like organisms. So if we could grow the same crops that we grow today, but without that damage and without those diseases, we would be able to feed an extra 4 billion people per year. It's quite staggering, the impact that these things have then on our food production systems. And so since we have this where at least 10% of all of our crops are destroyed by plant diseases, this also leads to an over-reliance on synthetic pesticides. And therefore, this is also very bad for the environment and, again, for human health. So the particular organisms that I'd like to introduce you to today are called Oomycetes. And these organisms, they look like fungi and they occupy the same ecological niches, but in fact, they are actually grouped in a separate kingdom of life, the Stremminopilla, and this is where the brown algae that live in the sea and a lot of marine organisms live. And these things are, have a wide range of ecological niches that they occupy. They cause diseases in major food production systems like salmon. They affect grapes, so they affect winemaking. In fact, this Oomycete disease here was the disease that allowed the discovery of the first ever fungicide, Bordeaux mixture. They have a huge impact on our environment and our landscape. And here you can see that there are tree diseases. Some of the Phytophthora are devastating tree diseases and are wiping out areas of our environment and parkland. And then we have potato late blight, which is one of the diseases I've been having a major focus on, which is hugely destructive. Other plant diseases, we also have tropical diseases. This is taro blight, affecting tropical plant production. And then we have organisms that are Oomycetes, but they, rather than eating plants, they are able to attack fungi or other Oomycetes. And so here we have a mycoparasite, that is Pythium oligandrum, and that is an organism that's able to feed on fungi and Oomycetes. And then we have also some that feed on animals such as mosquito larvae, like Lagnidium here. So I'd like to take you on a journey to discover some of the interesting biology and facts about these organisms and why we work with them. And I want to give you 3 different examples today: the good, the bad, and the ugly. And you'll see, I hope at the end, that the ugly is a little bit like the story of the ugly duckling, because although he's ugly here in his picture, in fact, if he can be used to wipe out mosquito-borne diseases, then I think you'll agree that would turn into a beautiful thing for humanity. So the first organism, the good then. What is good about the Oomycetes? Well, as I just told you, some of them are mycoparasites. And that means we can use them for biological control. And what do we mean by that? This is where we use an organism to influence or to suppress a disease-causing organism, in this case, plant pathogens. And so agents that can be used for biological control, BCAs, may be fungi, oomycetes, bacteria, or nematodes. And we often find them in the as natural inhabitants of the soil or the environment. And then, of course, we find them where they are not pathogenic to humans or to other organisms like birds, animals, or fish. And they can have different modes of action. Mycoparasites feed on fungi and other oomycetes. They can make antibiotics and therefore exhibit antibiosis. They can also compete for nutrients or availability of resources in the ecosystem, and they can induce resistance in host plants. And so some of the oomycetes that I'm working with that have potential to be biocontrol agents, the first one is Pythium oligandrum. This has a wide host range, so that means it will feed on a lot of different fungi, a lot of different plant pathogens. It also can boost plant growth and induce resistance. And it can kill its host quite fast, in 2 or 3 days. And it's actually registered as a biocontrol agent in Europe and in the US, and there is a commercial product available, Polyversum. We have a second Pythium species, Pythium periplocum, which has a narrower host range and takes a little bit longer to kill hosts. But this organism can survive much better than oligandrum in dry soils, so it may be useful to be used in some places where we have climate limitations. For example, I've just come back from South Africa, and in the Western Cape, as you know, they have been living under a drought condition since 2016. And so there's a real need there to find ways to protect crops under drought conditions. And then we have 2 species that are also able to parasitize the mosquitoes, and I will come back to these with our ugly pathogens at the end. So some of the scientific questions we ask are how the interactions between the host and the oomycetes shape their genomes, and how those genomes are shaped by those interactions, and how we can use that knowledge to improve the prospects for biological control using these organisms. So Pythium oligandrum is the one that I will focus on for now, which feeds on fungi and other oomycetes. And it's able to exhibit all of the modes of action I told you about as a biocontrol agent. So it feeds as a mycoparasite. It also secretes enzymes, toxins, antibiotics. And it's very fast growing, so it can outcompete other microbes in the ecosystem. But we need to know more about this biology and the genetics to really be able to use it more effectively. And so we're investigating that, and also the effects that this organism has on soil health by looking at, for example, the potato root microbiome when treated with this organism compared to synthetic pesticides. So we can try to see what is the effect on the community of microbes and how can we encourage that to be a beneficial community of microbes that keeps diversity and keeps plant protection naturally in the soil as much as possible. And then, of course, we need to know how this organism could actually work in practice, and therefore we're testing this organism to control different potato diseases. This is what Pythium oligandrum looks like down the microscope. You can see on the right-hand side here is a host, hyphae, and then you have coiling, like a boa constrictor. It's coiling around its host and eating the contents. At the beginning of the infection, it's making these little hooks. It senses the host and starts making hooks and then starts feeding. And these are the spores, the oospores, that it produces and that we need to use to make a formulation, for example, of this organism. So these spores can survive in the soil for many years because they have very thick cell walls and a high level of fat reserves. Just like an egg, they contain a lot of lipids ready for when it needs to germinate. And this is a scanning electron micrograph of an oospore. And you can see this is all just the cell wall here, and then here we have a lot of fat reserves. So what happens is these spores can become dormant, and if you would like to make them as a preparation for a farmer to use, you need to make them into a wettable powder that you could then use in a farm sprayer. And so when we dry them, they enter a dormant phase, and what we don't understand at the moment is how to wake them up effectively to make them germinate. How do they sense when they need to germinate and start becoming active again? So we have sequenced the genome of this organism, and we are studying the gene expression of this organism when it's producing these spores and when it's infecting different hosts, to try to understand what are the cues and what are the molecular mechanisms that cause it to germinate and to start becoming an effective mycoparasite. One of the things we've identified is that small tyrosine-rich proteins, they're very highly expressed during parasitism, but also during the formation of these spores. So what we've been able to do is silence those genes so that they are not able to be expressed anymore. And then you can see then that the O spore looks quite different from the normal spore here. We have very disordered cell wall, and in fact these spores cannot germinate anymore. So we can understand then that these proteins are very important for correct formation of the wall and for germination. We also identified lots of genes that are involved in signaling and are highly expressed during these phases. And we're now looking into those to see what can we manipulate or guide to help make a more effective formulation of this organism. What we can also see from the basic biology is that when we grow this organism, this Pythium, with a host, for example, Phytophthora infestans, that's the potato late blight disease that I will tell you about in a minute, we can see there's a dynamic battle going on in gene expression between these two species. So what happens is Pythium is expressing enzymes to degrade the cell wall of its host and a lot of molecules for signaling to allow it to activate its mycoparasitic and pathogenicity genes, and then is making toxins and also nutrient and drug transporters to then obtain its food from its host. But the Phytophthora has a response as well. It understands that it's under attack, and it produces enzymes to try to strengthen its cell wall, to build the fortress so that it can't be attacked. It also makes enzymes that inhibit enzymes that are produced by the mycoparasite. And expresses resistance genes and other things that it tries to use as counterattack molecules. So it's very interesting at a basic biology level that we can have this kind of attack and counterattack defense battle between two different microbes in the soil. But also we need to understand how these organisms can defend themselves, because if we want to use these strategies in future plant protection, we need to know that they will be durable and sustainable. And so by looking at what the host is doing, we can try to understand whether it will be able to develop resistance to a biocontrol agent or not. So now I'm going to move on to the bad. So here I'm going to talk about the potato late blight pathogen Phytophthora infestans. This is a very devastating disease, which originated in the Mexico and or the Andes regions. And it first entered Europe in shipments of gunpowder in the 1800s, and then it came over again in the 1970s when we had to get seed potatoes. And it's still a global threat today. It's been a huge problem for humanity. It's been described as one of the diseases that's had the highest impact on human society ever, because it caused the Irish potato famine in the 1800s, which spread also across the whole of Europe. And in fact, millions of people died and emigrated from Ireland at that time, and the population of Ireland has never recovered to the level it was before the outbreak of this famine. And it's still a big threat today. And famines can even be triggered still in modern times. In the year 2000 in Russia, there was a famine because of potato late blight again. And we don't have good, effective, sustainable controls. We have a lot of synthetic pesticides that we can use, but this means repeated spraying throughout the growing season, which is not so good for the environment, for biodiversity protection, or for the farmers. And so it's estimated that this disease can cost up to about 6 billion euros per year to treat worldwide. And you can see it has an effect on both the foliage of the plant, just destroying the plant, but also on the tubers. And this is how it looks like under the scanning electron microscope. So we're interested in really finding out, along with many other people in the research community, what the mechanisms are at the molecular level for this disease. And we have identified small proteins that are being secreted by Phytophthora that are used to attack the plant and to allow it to hide from the immune system that the plant has, and then also to obtain the nutrients. And these are called effector molecules. And if we can understand the function of these molecules and of the molecules that are essential for the growth of this pathogen, it will be very beneficial, because it will allow us to design much more durable control strategies. So we've developed a technique based on RNA interference, which allows us to silence several genes in Phytophthora infestans. So that is to stop those genes being translated into proteins. And then we can look for a phenotype in our cells to see then what the function of those genes are, because many of these effector genes are predicted from bioinformatics, but we don't know enough about what they do. And what we have done is then looked at key moments in the development and the disease cycle of our organism to try to find out what is essential for growth or what is essential for pathogenicity, and what can we therefore interfere with as in a control strategy. So what happens when, to start the infection, is the spore from the late-blight pathogen lands on a leaf, and here is the spore on the surface of a leaf under the microscope. And it germinates, it makes an apposorium. This is a specialized structure that allows the organism to penetrate the cuticle of the plant. And then it can go on and feed inside the plant. And this structure builds up pressure and secretes enzymes. And what we have discovered is that it's very important to have active synthesis of the cell wall carbohydrate cellulose at this point in development. Otherwise, the pathogen is unable to even start the infection. So what we can do at the molecular level is prevent the production of cellulose synthetic enzymes, and then you can see what the effect is here. The spore is unable to make this proper structure for infection, and it can never actually penetrate the plant. And this is such an important function for the pathogen that it's very unlikely that you can develop resistance to this kind of chemicals. So Syngenta has actually produced a fungicide now, mandipropamid, which targets cellulose synthesis in Phytophthora because we realized that this infection process was so important. You can see in the picture here, when you inoculate the pathogen onto a plant with fully functional cellulose synthases, it can cause infection, but on the other half of the leaf, when it does not produce cellulose, it's completely unable to infect. So we have a way to be able to develop more durable pesticides that can be much more targeted to the precise development of our pathogen. So now I'm going to turn a little bit towards the ugly. And here I'm going to give you a very short example of the mosquito pathogen Lagnidium giganteum. And this is a very interesting oomycete because it infects the larval stages of many mosquito species, including some of those that are vectoring important human diseases. And you can see that the mycelium eats the whole larvae in a very few days. And it's interesting because it makes very similar infection structures to plant pathogens, even though you would imagine that to infect a mosquito larva or a plant surface would be rather different. And it's been registered as a biological control agent in the USA. What's also very interesting is that some of these subspecies of this organism have been found very recently as a human pathogen, particularly in Thailand, where this organism likes to live in tropical conditions. And so what we can see is that oomycetes are very adaptable, and they can make host jumps. And here we have an example where an organism has developed an ability to go from mosquito to humans, and you can get very nasty ocular infections with this organism. So it's very interesting from a basic science point of view to understand why that can happen and what is going on in those in the genes and the evolution of this pathogen to allow it to suddenly be able to make host jumps. And so we have a reference genome that we have assembled, and we are looking at gene expression in this organism. And we're comparing that to the plant pathogens and to the biocontrol agents to get a better understanding at the genetic level of what makes each organism target a different host, and then how we can better manipulate those organisms for our own benefit. Another one of the ugly pathogens is cocoa black pod disease, and these are invasive species that have just moved into West Africa and having a big threat to our cocoa production. It's a huge global market for cocoa, of course, and we now have both viral diseases and new oomycete diseases that are attacking our cocoa. And so one of the other projects we're working on, as well as potato, is then to look at how we can support farmers in West Africa in dealing with these invasive species, how we can provide them with better knowledge, molecular tools to diagnose and to treat these diseases so that we can boost their production in this very, very poor region of the world. For just last couple of minutes, I would like to talk a little bit then about how we should take this basic knowledge and put that to use in integrated plant protection strategies. So what we're trying to do in my research group, and then in combination with many different researchers, both here and in other parts of the world, is to take a multidisciplinary approach with many different methods to integrated plant protection or pest management for potato and cocoa. And so we have colleagues working with remote sensing, trying to use drones or satellite images to identify when the disease starts in the field. We take our genomics knowledge, we take ecology knowledge with colleagues that are working on how these disease organisms can overwinter in wild relatives of our crops. We look at the soil health and the microbiome. How is the microbial community affected by disease or by different preventative treatment strategies? We work also with disease modelers and with modeling to help farmers have good decision-making processes for when they should spray, if they need to spray. And we would like to try then to reduce the use of synthetic pesticides and increase the use of low-risk inducing agents, biological control strategies, and working both with resistant cultivars and with boosting the plant's natural immune system. We therefore hope to then achieve more integrated protection of crops like potato and cocoa. And so what we've started to do is test some of our things in the field. And so here we have tested different treatments, silicates, bacterial biocontrol agents, and our new Pythium species to see if it can have an effect at controlling early blight disease caused by Alternaria solani in potato in the field. And what we can see is we get a little bit of protection with our Pythium oligandrum, but it's not as effective as the most effective control treatment. The untreated is the blue line between here, so there's not a statistically significant difference. So we know that biological control agents cannot on their own replace pesticides. But therefore, I think we need to have combination strategies where we combine different biocontrol agents and low-risk compounds to try to get more sustainable disease control in combination with using resistant cultivars. And so that's what we have started now to look into, is how to make combinations between these different control treatments to be more effective. And so we're using also starch potatoes here that have a little bit of resistance. And so now you can see here is our disease curve from our untreated potato, and then here we are using either single or combined compounds. And in this case, when we combine a bacterial biocontrol agent and our Oomycetes control agent, this pink line here, we have a more significant level of disease control. So I think this is very good for the future, that we're going to be able to work more with combining these things and not thinking just about single genes, not thinking about single resistance genes or single chemicals, but actually thinking about a holistic approach to disease control. So this then is a little bit of a summary of what we're working on and trying to achieve for integrated plant protection for both potato and cocoa. We need to better understand the basic biology and the genomics of our mycoparasite and its interactions with its hosts, and this is going to aid in our formulation and application of these kind of agents. And we need this integrated approach where we reduce the reliance on synthetic pesticides. We maybe don't take them away altogether. Maybe we sometimes need, when we have a high disease pressure or early on, to make some treatments, but that's why we bring in then multiple strategies and decision support systems to make the farmer as informed as possible so that we really can reduce that reliance rather than taking it away altogether, and integrate these different approaches. So a treatment strategy might involve spraying 1 week with a biocontrol agent, 1 week with a low-risk compound, 1 week with a pesticide. And during this process, we also need to do research and have good communication on how we minimize the impact of these treatment strategies on the ecology of the microbes that are in the communities in our fields. And therefore, we then need to connect this basic science with practice, with how we carry out our control strategies. And again, rather than looking at a single disease like potato late blight, I gave you an example now, but we need to also look about the interactions with multiple disease agents or other pests in the same host. For example, we have both late blight and early blight in potato. How do they interact? Do they cooperate with each other, or do they compete in the field? And how will this affect disease control strategies and yield? And through this, we hope to then develop holistic approaches for disease control in these kind of crops. I started with some thank yous, and I will finish again with some thank yous, this time a little bit more scientific thank yous. I couldn't have achieved this also without the help and support of my fantastic team, the PhD students here in blue and the postdocs and researchers here in black. and with collaboration with all of my colleagues in plant protection biology. There's too many here to list all of them. Some particular recent interactions and support from Eric and Åsa and Erland have made much of this research possible, as well as important contributions from master's students and our collaborators across the globe and our funding agencies. And lastly, thank you all for listening.